Okay, good morning, everyone. Myself is Dr. Santosh. I am heading genomics. I think everyone has joined, right?
Yes, sir. Good
Yes,
morning.
everyone has joined. You can
Okay,
continue.
okay. Today's session will be on NIPT. This is called non-invasive prenatal testing. So let us understand this test in detail. And before starting in detail, let us understand these words first. Non-invasive prenatal testing. There are four different words in this test. So let us understand this. So non-invasive means without any invasion or without any entering anything in the body. Okay. So this is called non-invasive, and the other two terms are called prenatal testing. So this prenatal is a testing is a word used for the fetus which has not born yet. So this is our testing for fetus which is still not born, and we are testing their genetic predisposition. So I think the word has cleared to everyone. Non-invasive prenatal testing. So let us start. So actually, non-invasive prenatal testing, they are sometimes also called NIPS or NIPS or NIPD. So means non-invasive prenatal screening or non-invasive prenatal diagnostics. So basically, this test is a screening test, and this method is basically this test is basically done for pregnant women to check whether their fetus is predisposed to any genetic condition or not. So basically this test is done for no uh, pregnant women, and this test is also advised for a pregnant woman who is actually more age, like more than 35, 30 or 35. So this test is recommended for them also. So why this test is asked for them for a pregnant woman more than 35 years of age, because with age uh, the chances of getting chances of getting those predisposition towards genetic condition increases. So basically this test is done in between 10th week to 24th week of pregnancy, right? And this test actually doesn't harm any no mother and kid, because in this test we used to check the blood samples drawn from the venous of the mother or the woman. Okay. So from the blood we can determine the predisposition of fetus inside the uterus. So how actually this test works, like during pregnancy after 10th week, uh, so we all know, like DNA we all know, right? So DNA resides in nucleus, basically nucleus, right? So this DNA actually when we are testing the for the fetus, so during pregnancy a fragment of DNA is 200 base pair, 150 to 200 base pair fragmented DNA from the uterus, which is actually a origin of the fetal DNA, comes into the actually it is actually coming into the mother bloodstream. So this is that fragmented DNA, or they are also called cfDNA or cell-free DNAs. So we are basically testing this cell-free DNAs, which is basically a origin of fetus. So that cfDNAs is actually can tell you the predisposition of a fetus towards any genetic conditions, right? So the DNA that cfDNA, we are actually testing that cfDNA of fetus for any particular genetic conditions. Up to this point, I think we are clear, right?
Yes, sir.
Okay. So this test is basically
एक
done
मिनट
for
डॉक्टर संतोष जी, भैया दोस्त है, एक मिनट संतोष जी। हम लड़के बहुत सारे नए ज्वाइन हैं जो नॉन साइंस और फ्रेशर्स है, तो आई जस्ट वांट देम टू आंसर। ये तो देवेंद्र आंसर कर दिया, उसको तो पता है, एनआईपीटी भेजता ही है। सो आई
okay.
जस्ट वांट टू नो फ्रॉम अमित दिल्ली, अमित, अमन, आनंद, राजेश फ्रॉम कोच्चि, व्हाट्स ऑल, हैव यू ऑल अंडरस्टूड और नॉट?
So if you people have
Yes,
not
yes, yes.
understood, I will start from scratch.
Morning, sir. It's clear, sir.
या मॉर्निंग यार, देखो ये सेशन बहुत इंपॉर्टेंट है गाइस, please understand the importance of this session. Lab is spending a lot of time for you. You have to utilize. Please ask anybody who is presenting. If you don't understand anything, you just please frankly speak to them, talk to them, and ask them. Make sure that you understand better, so you will sell better. So that is the point here. So please do not hesitate. I'm keenly checking all the marks of everyone after this sessions. So I want all of them 10 out of 10. So please ensure that you spend a lot of involvement in the meeting, okay? Training. ओके डॉक्टर संतोष जी, जरा ना नए लड़के बहुत हैं, थोड़ा सा आप पर्सनली एक एक से पूछ लिया करिए। अगर आपको लगता है कि ये लोग आंसर नहीं कर रहे हैं, तो जवाब नहीं दे रहे बहुत देर तक, दे आर नॉट आस्किंग एनी क्वेश्चंस, मेक देम आंसर, ओके?
Okay. So, for non scientific background or non-science candidate, so let me explain in other way also, other way around for this first slide. So, actually, any genetic test needs DNA. So, the source of DNA will be chromosome, right? And the chromosome resides in the nucleus. Okay, they are called genome. Okay, so let us start reverse. So cell contains nucleus, then nucleus contains genome. Genome is made up of chromosome, and each of the chromosome, they are made up of chunks of exon, introns, like that, and each of those exon and introns are further made up of nucleotides. Okay, understand? No? Hello.
Yes, yes.
Yeah. Okay.
Yes,
Fine.
Santosh.
Yeah. So to understand the genetic predisposition or genetic disorders, you can say, okay, or genetic disorder, you can say, if a fetus is predisposed to, or like in future, if this particular fetus born into a child or a kid, so they may not contain any genetic disorders, right? So this test is just a screening test at the initial stage of pregnancy to avoid those kind of kids to take birth, because that kind of birth may be a problematic to their parents also and the kids also, because they are not genetically fit and they cannot mix with the society, right? To avoid this kind of kid in actual in actual world, we are actually doing this test to eliminate those kids at the pregnancy level. Understand? Hello, Ram is there.
Santosh, can you repeat the last sentence what you told?
Yeah, to avoid birth of defected or defective kid. Okay, so this test is a screening test of a pregnant woman between 10th week to 24th week, because after 24th week it is illegal to abort a child, right? So this test is done in between 10th to 24th week. Okay, hello, understand?
Yeah.
Yes, understand.
Yes,
Yeah,
Santosh.
please no just tell me yes or no, so that I will skip to other slide, or otherwise I will stick to the same slide, make you understand. So the uh, the common predisposed genetic conditions are that will be explained in next slide, but I've abbreviated in this slide only. Like T21, this is actually T refers to trisomy, right? So trisomy 21, trisomy. This I will explain you what is trisomy, right? You will be visualize this trisomy in next slide. So trisomy 21, trisomy 18, and trisomy 13. Beside that, due to PCPND Act, we cannot determine the sex of a child. So XY are not reported in this text, right? So let us understand further. So this is actually a NGS based test. So how many of you know NGS?
Next generation
The
sequencing.
next generation sequencing.
Next generation sequencing, correct. Okay, fine. Means some of them are aware of this next generation sequencing. So let us understand this from scratch. So what I explained is like chromosome is the basic source of genetic testing, right? Or genome is the basic source of genetic testing. So NGS is a technology. This is a parallel sequencing technology. I mean, it's a mass sequencing technology from where we can determine the sequence. Okay, from the sequence we can determine whether particular thing is present or not. Okay, particular genetic condition is present or not. Is a normal or a diseased condition, right? So this is technology which we are using for NIPT. Okay, so the system or the instrument which we are using for this NIPT testing, okay, can vary. Okay, like Illumina, we can use any of the sequencer like 500. We can use Illumina 500. We can NextSeq 500. We can use, okay, HiSeq we can use, right? So we can use those instrument to assess this test. Okay, so we'll come to the next slide. If first slide is clear to everyone.
Okay.
Okay, so anyone need to understand this pictorial representation over here? If anyone want to understand this, I will make them understand this pictorial representation also. So why we are doing this blood test and what is NIPT again? Then see the figure represent. This is a placenta, right? So this is a placental DNA. What I have explained you, like these are the 200 or 150 to 200 base pair fragment of fetal origin DNA. Okay, that actually comes into mother bloodstream after 10th week of pregnancy. So the test is performed after, I mean, from the 10th week to 24th week, right? So this is the source, this placental DNA, or they are also sometimes called cell-free DNA because they are free from the original cells, right? So this actually comes in the bloodstream. From this bloodstream, we used to collect the blood, right? While collecting blood, this fetal DNA also comes in the sample. So this sample we are testing. Okay. Okay or not?
Yes, yes.
Yeah.
Yes, sir.
Please help me in saying yes or no. Okay. So as I explained in the previous slide, like these are the three basic chromosomal condition we are referring in NIPT. So these are the condition. Just look at the born child. What is their condition? See, first one is trisomy. Okay, trisomy 13. This is called Patau syndrome. So just see, 13th chromosome has triplicated, right? One extra chromosome has been added here. Can you see the marking of trisomy 13? Right. So see the condition of the child. This is not a normal child, right? To avoid this kind of child, we are testing. We are into this NIPT. Understand? The other kid is this trisomy 18. See, this is called Edward syndrome. So just see. There is a triplication of 18 in the 18th chromosome. So this condition arose. So the other case is trisomy 21. This is called Down syndrome also. In all of these three tests, and somehow the child is mentally disturbed, physically disturbed. So this condition will lead to the um, parent cannot take this, no. So to avoid this, we are doing this test. So let us understand is prevalence. Prevalence means what is their percentage, or what is their occurrence in a general population. Just check, we'll check it. So incidence of this chromosomal disorder is actually in India. It is not India, in entire world. It is actually 1 out of 200 birth. 1 out of 200 birth is happening because of non-testing those no non-testing of those pregnant women. So one doesn't represent a single number. It is actually in 200 births, it actually represent one case where any of the three cases can happen. Understand?
Screen is
See,
not showing. Sorry,
I'm sorry.
screen is not coming.
Hmm?
Screen is not coming, sir. I think.
Now?
Okay, fine. Fine, guys. This actually is Dr. Santosh, एक मिनट दीजिए. I'll just check out. मेरे को कुछ मीटिंग में जाना है. Just I want to talk 2 minutes and I'll leave. So, guys, this importance of NIPT to detect any abnormalities in baby at the time of pregnancy, to identify. We will do this NIPT, non-invasive prenatal testing. This is full form of NIPT, and it will be done only to identify any abnormality fetus. To at 15 to 20 it's ये टेस्ट करते हैं normally 14 weeks, 15 weeks till 20 weeks we can do it to identify and take a decision. सो इसके अंदर जो डॉक्टर संतोष जी समझा रहे थे, सिंपल लॉजिक में मैं समझा देता हूँ कि ये टेस्ट में मदर का ब्लड लिया जाता है. देर आर 2-3 प्रोसीजर्स एक्चुअली. अगर प्रेग्नेंट वीमेन में अगर हमको प्रॉब्लम फाइंड आउट करना है, इफ यू वांट टू फाइंड आउट एनी क्रोमोसोमल अबनॉर्मलिटी और एनी डीएनए डिफेक्ट है, तो वी कैन easily identify on this technique called NIPT, which we do on thermo sequencer. ठीक है. एनआईपीटी में मिनिमम क्रोमोसोम्स करते हैं, वो सारे टेक्निकली बता देंगे. इसके लिए मदर का ब्लड लिया जाता है. मदर के ब्लड में से फीटस का डीएनए सेपरेट किया जाता है. उसको बोलते हैं सेल फ्री डीएनए, जो डॉक्टर संतोष अभी समझा रहे थे आपको व्हाट इज सेल फ्री डीएनए. तो मदर के ब्लड से जो सेपरेट करते हैं उसको बोलते हैं सेल फ्री डीएनए. उस डीएनए को टेस्ट करते हैं, उसके क्रोमोसोम्स टेस्ट करते हैं. अगर उसकी लेवल्स नॉर्मल और उसके वो होते हैं ना, मतलब उसकी जो लेवल्स होती है, अगर नॉर्मल एलिवेटेड होती है, नॉन एलिवेटेड होती है, वो हम दिखा देते हैं. इसमें बताते हैं रिस्क कितना है, अगर ये कैरी करना है आगे नहीं करना है, वो रिस्क फैक्टर बताते हैं ये टेस्ट से. एंड दिस इज 99% एक्यूरेट टेस्ट टेक्नोलॉजी. इट इज नॉट 100%. ओके, 100% दूसरा प्लेटफॉर्म है. एनी हो तो बाकी ये समझा देंगे, आप आराम से उनसे बात कर लेना. डॉक्टर करो, आई एम जस्ट
Yeah. Okay, sir. Okay, sir. Okay, sir.
फॉर अ
Okay, sir.
मीटिंग. ओके.
Okay, sir. Okay, sir. Okay. So, everyone can see, no, this third slide, NIPT
Yes,
prevalence.
sir.
Okay. So I was explaining, no, the consequences if we are not getting tested for this test, right? So these are the three consequences I've shown you for NIPT if we have not done, and the prevalences I will explain you. Okay, so uh, there is a prevalence if this test is not performed. So this may happen. Okay, this kind of child may born. They are mentally retarded. Somehow they are not physically well. So to avoid this time condition, we are doing this test. Okay, so to whom this test is no ordered, as I explained in the previous slide, in the first slide, like pregnant woman. Okay, can any pregnant woman can go for this test, right? But this test is particularly carried out for a woman who is more than 35 year of age, right? Why we are saying is like that, because with the age, okay, the with age, this genetic condition of this egg is changing very fast, right? So that chromosome not remains stable in that condition. So we are actually recommending this test for this NIPT test. So if any woman have a prior history of pregnancy with trisomy, I'm saying like no, like trisomy, any trisomy, like 18, 21. Any case, if this lady has suffered a trisomy case previously in the previous test, right? So this test has is recommended for them. Okay. Or any pregnant woman showing this ultrasonographic finding indicating increased risk of aneuploidy. So ultrasonographic finding, actually, ultrasonography tells about the feature of the facial feature of the kid. That facial feature actually reflects the genetic makeup of the child. So these are the different condition where we are telling a pregnant woman to go for NIPT. But uh, universally speaking or generally speaking, this test can be taken up by any pregnant woman to avoid those kind of predisposition. So up to this, clear?
Yes, sir.
Yes, sir.
Any question in this slide, please ask me.
Sir, when we will start NIP? As per ACOG, I believe it is 14 weeks, but internationally, if we are going, then we might start it after 10 week only.
Yeah. So, what I explained in the previous slide, like cfDNA or cell-free DNA start accumulating in mother bloodstream at the age of 10th week, right? So, we can test pregnant women from